Al has recently learned that if the ends don't have to look for each other, but instead are connected by a piece of plastic elastic tubing, rats can, like tadpoles, replace up to 3 centimeters of missing intestine. There's no reason to believe this technique couldn't be adapted to humans. Even though we don't know enough yet to electrically stimulate intestinal healing, Al has proposed a preliminary test of regeneration in large mammals that could spare some patients a lifetime of misery. It's almost impossible to surgically rejoin the colon, and when sutures fail, the person ends up with a colostomy. Since the free end of the colon would be held near its proper position by the local anatomy, Al suggests replacing it without stitches and giving such animals a temporary colostomy upstream from the cap. If x-rays later showed enough regrowth, the temporary colostomy would be closed and the animal would have a continuous healthy intestine. If even a few patients could be spared the indignity of living attached to a bag, the effort would be well worth the little, yet still non-existent funding required. Intensive study of the electrical details of gut healing would probably make surgery less devastating for many additional patients. Exciting as prospects in this survey are, they're by no means the only ones or even the most spectacular, which are reserved for the following chapters. Many researchers are working to turn the breakthroughs of the last 2 decades to practical use. Even so, progress isn't nearly as fast as it could be. Perhaps due to disbelief, such widespread self-repair is really possible for us. It's not only possible, it's nearly certain, given even a modest monetary push for the useful dispositions foreseen by Spallanzani are within our reach. Chapter 10, The Lazarus Heart. Like Columbus, scientists sometimes stumble upon new continents when merely seeking a quicker made route. Our research group had this good fortune in 1973. We'd gone back to the basics after learning how to differentiate frog red blood cells and start regeneration in rat limbs. We decided to study nucleated red cells in a variety of creatures, hoping for leads towards better regrowth in mammals. Although their circulating erythrocytes have no nuclei, even mammals have young red cells with nuclei forming in the bone marrow. After severe bleeding, up to a fifth of those in the bloodstream may be immature nucleated types, as the marrow rushes them into service to make up for the loss. We surveyed the effect of direct current on red cells from fish, amphibians, reptiles, and birds. All of the cells responded, but in a different way for each species. We decided to have more detailed look at the largest and hence most easily studied red blood cells available. Those of our old friend Tritarius medicens, the common green newt. A newt is so small that you can't just poke a needle into one of its veins and take a blood sample. The only practical way to get pure blood is to anesthetize the animal, slice open its chest, and cut its heart into extract the blood with a pipette and throw away the carcass. As the phrase goes, we harvested blood from 3 newts each week by this method. One day, when Sharon Chepin had finished the chore, she asked me, "What would happen if I sewed these animals up?" I answered that because their hearts had been destroyed, they would die within minutes, with or without sutures, from the lack of oxygen to the brain, just as all our other amphibian blood donors had. We looked it up just to make sure. The standard works on regeneration all agreed that no animal's heart could repair major wounds. Unlike skeletal muscles, the cardiac variety had no satellite cells to serve as precursors for mature heart muscle cells. In any case, the textbook stated the animal would die long before such repair could occur. Next week, Sharon put other 3 intended sacrifices in a bowl of water and with a straight face asked me if they looked healthy enough to use. I told her they looked fine. Good, she explained. These are the same 3 we used last week. Score 1 point for the open mind. Flabbergasted, I helped anesthetize and dissect 2 of the miracles. Their hearts were perfectly normal, with no evidence of ever having been damaged in any way. a 5-alarm blastema. Abruptly, I changed my research plans. I asked Sharon to test a series of newts by cutting away large sections of their hearts and sewing up their chests, then killing some of the survivors every day and slicing, mounting, and staining the hearts for study under the microscope. Over 90% lived through the first operation, and several weeks later, we had hundreds of slides ready for examination by now. We knew we had come up with a first-class mystery and had better jettison our preconceptions. We reasoned that we could tell when regeneration was finished by finding out when the blood began flowing again. While waiting for the specimens, we rummaged more thoroughly through the literature for other reports on heart regeneration. There was evidence for limited repair, but no true regeneration. Small heart wounds in a few animals. The process seemed limited to the very young. Even then, the results were of poor quality, combining a lot of scar tissue with only a little proliferation of nearby heart cells. But the two mitotic component could be enhanced by various experimental aids. In 1971, John O. and Jean Oberpreiler, anatomists at the University of North Dakota School of Medicine in Grand Forks, reported that small wounds in salamander hearts healed this way, but required 2 months. A year after that, the English edition of a book by Lev Polichev summarized several decades of Russian research, mainly on the hearts of frogs and lizards. Pavel Rumyanstev, now at the Cytology Institute of the USSR Academy of Sciences in Leningrad, had found in 1954 that newborn mammals, rats and kittens, could repair tiny puncture wounds, and recently he has proved the same capacity in the atria, or receiving chambers, of adult rat hearts. We even found a German report of 1914 claiming that human babies had sometimes regenerated small areas of their hearts damaged by diphtheria. The Russians claimed that some progress in extending this marginal native healing. In the late 1950s, N.P. Sinistin had repaired large holes up to 16 square centimeters in the hearts of dogs by covering the wounds with patches made of muscle sheath, canvas, suede, and other materials. Scar tissue still covered the outside, but the patch guided a thin layer of new muscle fibers forming along its inner surface. Using dogs whose wounds had already closed with this scar tissue, Polichev then found he could induce heart muscle to fill in part of the gap by cutting away the scar and irritating the edges of the remaining cardiac muscle. Other Soviet researchers enhanced the muscle cell proliferation a little more with vitamins B1, B6, B12, various drugs, extra RNA and DNA, and the heart tissue extracts, or menses. Heart regrowth was limited to very small injuries or the border zone around larger ones, and it always took several weeks. No one had even imagined that half a heart could restore its other half, much less in a matter of hours. I could hardly wait until the next batch of slides was ready. They showed us an unprecedented type of regeneration. Where the missing part of the heart had been, a blastema formed in about 2.5 hours. We saw no evidence of dedifferentiation or mitosis in the remaining heart muscle cells, and indeed it would have been impossible for the processes we'd already studied to make a blastema in such a short time. Instead, the mass of primitive cells arose dramatically from the blood. As soon as the salamander heart is cut open, blood pools around the wound and clots quickly, usually in about 1 minute, sealing the hole like wet plaster. Almost immediately, the nearest red blood cells crack open like eggs. Their nuclei, surrounded by a thin coating of cytoplasm, glide by some means yet unknown directly to the raw, frayed edge of the heart muscle and insinuate themselves into the tangle of dying and injured cells. To a biologist, this sight is bizarre, uncanny. It's as though the engine of a passing car could walk up to a stranded truck, climb under the hood, and drive it away. Super regeneration, the salamander heart. Farther away from the wound surface, the red cells also spill out their nuclei, but these cells yolks clump together, fusing the remaining cytoplasm to form a syncytium. Still farther away from the center of action, the red cells undergo more leisurely dedifferentiation we observe in our frog fractures and DC culture studies. They turn into primitive amboid cells that move toward the area of damage and attach themselves by pseudopods to the injured muscle fibers. In all of biology, there's no precedent for these virtuosic cellular metamorphoses. In fact, they're so strange that the most researchers have simplified, refused to believe in their existence or try the experiment for themselves. All these changes are well underway within 15 minutes. Soon afterward, the extruded nuclei are interconnected syncytial nuclei, and the amboid cells are divided as fast as they can, building up the blastema. It's fully formed within 3 hours after the injury. By then, its cells have already started to redifferentiate into new heart muscle cells, synthesizing their orderly arrays of contractile fibers and connecting up with the intact tissue. If the color contained more red blood cells than we needed, the extras outside the area now degenerate, apparently so as not to get in the way of the repair work. Meanwhile, the new has survived by absorbing dissolved oxygen from the water through its skin. Now at the 4-hour mark, there are enough new muscle cells without there are enough new muscle cells to withstand contraction, and the heart begins pumping again slowly. After 5 or 6 hours, most of the blastema cells have redifferentiated into muscle, which is still somewhat lacy or delicate compared with the established tissue. After about 8 or 10 hours, however, the heart is virtually normal in appearance and structure, and after a day, it's indistinguishable from an uninjured one. Why did we see this colossal regeneration while the other prillers found only a tiny, slow healing response in the salamander's heart? Apparently, this was another manifestation of Polichev's principle. We made a big wound, they made a small one. Only massive damage unleashed the full power of the cells. Is this fantastic cellular power forever restricted to salamanders, or does it reside latent in us, ready at the appropriate impetus to repair damaged hearts without problem-filled transplants of donated or artificial pumps? We don't know, but when we found no other regenerative process that forever off limits to animals, at this point, we could only speculate on how such a treatment might be accomplished, but at least the idea isn't wholly fantasy. The first job is to identify human target cells able to dedifferentiate into primitive totipotent cells. Bone marrow cells are immature erythrocytes, the nearest equivalents to amphibian nucleated red blood cells, are one obvious candidate population, especially since they seem to be the crucial cells in rat limb regeneration and the inner part of fracture healing. Fibroblasty specialized bioelectrically injected silver ions might be used. Another possibility is lymphocytes, one class of infection-fighting white blood cells. In our lab, we've demonstrated that they too can dedifferentiate in response to appropriate electrical stimuli. Since new type heart regeneration doesn't occur naturally in mammals, we would probably have to grow a large mass of target cells in tissue culture. Then, with the patient on a heart-lung machine, the surgeon could cut away scar tissue and otherwise freshen the wound if it wasn't recent enough, then apply enough of these ready-made pre-blastema cells to fill the defect. They would be held in place by a blood clot, sutured pericardium, or some type of patch. Then, assuming we'd learned the electrical parameters already, electrodes would induce nuclear extrusion, dedifferentiation, consolidation with the surrounding muscle, and the final transformation into normal cardiac muscle. The current would probably have to be adjusted through the process to get its various steps in synchrony, and vitamins or drugs might be used to enhance mitosis or protein synthesis once the scars had been removed. The instructions as to what cells were needed would come from the surrounding healthy heart muscle. Possible regeneration of the human heart. In the salamander, this process takes about 6 hours, since this exceeds the current of machine time for artificial and circulation in humans. We would have to extend the capacities of heart-lung devices or else speed up the cellular processes. Obviously, there's a long road of experiment to travel before we can be more specific about techniques. One of the things we must learn is whether the Newt's electrocardiogram shuts down during repair. We must know how its presence or absence relates to the current of injury and other electrical factors of this novel method of blastema formation. Personally, I'm sure we can get the human heart to mend itself as a result of being confronted by this wonder in Newt's. I'm convinced that the potential repertoire of living cells is absolutely enormous, far greater than the healing powers of normally manifested by most animals or even those dreamed of by doctors. Even the Newt in this super regeneration doesn't appear unless 30 to 50% of the heart is gone. Something about the massiveness of the injury or the approach of the death then boosts the healing process into overdrive. I readily admit that the discovery sounds a bit like science fiction, even as toned down into the subdued technical prose of our report. Published by Nature in 1974, I had trouble believing it myself at first because it seemed so incredible. There was no rush to confirm and extend our discovery. Today, even our observations have been corroborated by the University of Michigan anatomist Bruce Carlson in 1978 and by Phil Person in 1979, complete with electron micrographs of the cell changes. Most biologists still don't accept heart restoration as fact. Perhaps because the reality is so outlandish, Carlson wouldn't publish, and Person has been unable to get his work published in the peer-reviewed journals. Our original paper of 10 years ago is still officially unconfirmed, and the other workers are still puttering around with little wounds. This attitude must change. Knowledge about the controls of this process will be incalculable value to medicine. For this is ideal healing. Spilled blood closes a wound at the body's center and replaces the missing part in a few hours. You can't get much more efficient than that. Chapter 11, The Self-Mending Net. Spinal paralysis is the most devastating of injuries and also one of the commonest. It afflicts over 500,000 Americans, including 15,000 new sufferers every year. Until recently, their outlook was absolutely bleak, for the human central nervous system had no known regenerative capacity whatsoever. Only a part of the spinal cord remained unsevered, with some recovery possible with physical therapy. Now, however, there is hope that we'll soon be able to coax nerve cells into reestablishing the proper connections across the damaged section, and thus return the use of arms, legs, sexual excretion organs, respiratory muscles, and the sense of touch to quadriplegics and paraplegics. In one way or another, this dream involves making human nerve cells behave more like those in simpler animals. The neuron is the basic unit of all nervous systems. It consists of a cell body containing the nucleus and metabolic organelles surrounded by dozens of filaments that carry messages in and out. The incoming dendrites predominate in sensory neurons. There's usually only one motor fiber or axon which carries the neuron's outgoing messages to dendrites of other neurons or receptors on muscle or gland cells. An axon, often several feet long, is the principal fiber of a motor neuron, which relays orders from the brain or spinal cord to the tissues and organs. All neuron cell bodies reside in the brain and spinal cord. Only their axons and dendrites extend outward, forming the peripheral nerves that connect every part of the body with the CNS. Other fibers connect certain sensory and motor neurons within the spinal cord, creating reflex arcs like those that jerk our hands from hot stoves without having to send the impulse all the way to the brain for instructions. Still, other fibers connect spinal neurons with those in the brain, and in the brain itself, the interconnections reach such a density that each nerve cell may hook up with as many as 25,000 others. The peripheral nervous system. Except for a few specialized components like the naked fiber tips that enter into the neuroepidermal junctions, all parts of the neuron are swaddled in various types of perineural cells. In the brain, there are several kinds collectively called the glia, in which the neurons are embedded in like hairy raisins in a pudding. The cell bodies in the cord are also surrounded by glial cells, but their axons and dendrites, which include the fibers of the peripheral nerves, are surrounded by Schwann cells. These form tubes made up of spiraling layers of membrane rich in fatty substance called myelin around some of the largest fibers. A third type, ependymal cells, line the four cavities within the brain, or ventricles, and the narrow central canal that runs the length of the spinal cord. These cells are close relatives, having all developed from the same part of the ectoderm, or outer cell layer, that form the primitive neural tube in the embryo. The nervous system actually consists of several times more perineural cells than neurons. Schwann cell sheath around a peripheral nerve fiber. Until recently, the perineural cells were considered merely a packaging tissue whose only job was to insulate and support the neurons. We've now learned that they play a major role in getting nutrients to the neurons. They also help control the diffusion of ions through the nerve cell membrane and hence regulate the speed of impulse firing, even to the point of inhibiting seizures, the random spread of impulses in the brain. They may also have an important part in memory, and they probably conduct the direct currents so important to regeneration. They're essential to healing wherever it occurs in nerve tissue. Peripheral nerve fibers can regrow. Otherwise, we'd lose sensation whenever we cut a finger. But neurons and their fibers in the CNS cannot. Peripheral nerves cell body survives safe in the cord or brain, and the cut end of the attached part of the fiber is sealed off. The outer severed part dies and degenerates. Some of its Schwann cells digest it, along with the now useless myelinated membrane layers. The empty Schwann tube remains, however, and begins to grow toward the proximal fiber, the one near the center of the body, whose Schwann cells are also growing across the chasm. When these cells meet, the nerve fibers grows along its reconnected sheath and eventually makes contact with the same terminals it's originally served. In salamanders, this process is very efficient. The Schwann cells can cross large gaps. An experiment who wishes to work with denervated limbs must be diligent to keep the nerves from re-entering. In humans, the two ends of the tube usually can't find each other over a distance of more than a centimeter. In that case, the proximal sheath with its intact nerve fiber hunts for its opposite number by growing in an ever-increasing spiral, apparently searching for some signal from the distal end, a part farther from the body center. Since each nerve is formed of many fibers, these spiraling tubes entangle each other in a lump of nerve tissue called a neuroma. Neuromas are painfully sensitive and often must be cut away. Occasionally, a surgeon can move the two ends of the nerve close enough for the Schwann cells to make contact. If that's impossible, the gap may be closed with a piece of nerve grafted from a less important peripheral nerve that can be sacrificed. Unfortunately, nerve grafts don't take reliably, and other methods, such as making artificial channels with tiny plastic tubules, are still in experimental stage. We don't know why the salamander's peripheral nerve regrowth is so much more effective than ours, but I surmise that its more efficient DC electrical system accounts for the difference. If the locator signal is electrical, it should be possible to augment it in humans so as to grow nerve fibers over long distance. Beginning with a 1974 report from David H. Wilson, the leads of general inference in England, there have been some interesting claims that pulsed electromagnetic fields have speeded recovery of limb function in rats after peripheral nerve damage, but the effect hasn't yet been substantiated for humans. If these findings hold up, we may soon be able to boost nerves past their 1 centimeter limit, even if the action is indirect, and a thorough investigation of the electrical basics could drive nerve regrowth to even greater lengths. The spinal cord. A sad and crucial difference separates peripheral fibers from those in the human spinal cord, for the latter don't reconnect over even a fraction of a centimeter. However, in the most injuries related, few of the neurons themselves are killed. It's important to realize that most of the cord cells below the injury don't die. The reflex arcs remain intact. In fact, reflexes are stronger than normal because the neurons are now disconnected from the regulating influence of the brain. For the same reason, the broken bones of paraplegics heal in half the normal time, whereas a bone will heal very slowly or not at all if its peripheral nerve supply has been cut. Only the communication between brain and spine is silenced in paraplegia and makes all the difference. Spinal fibers do reconnect in some animals, notably goldfish and, as you might expect, salamanders. Their ability seems to decline dramatically with age, however. Gerald Bernstein, a neurophysiologist now at George Washington University Medical School who has studied goldfish spinal regeneration extensively, has found that 1-year-old fish heal almost all of the damage. The competence declines about 70% at 2 years and 50% at 3. Since salamanders aren't raised in biological supply houses, but rather collected from the wild, any group is likely to include young and old individuals, making comparison difficult. In our lab, we found that cord regeneration isn't uniform in salamanders, probably due to age differences. Maturity may reduce it in the response of the epidermal cells, which are responsible for the first step. They proliferate outward from the central canal and bridge the gap in a few days. Mark Singer, in a recent study of this process, concluded that the epidermal cells extend arms and radiating outward, which line up like those spokes of wheels stacked upon one atop another, forming channels from the regrowing fibers to follow. The nerves then reestablish their continuity within a few weeks. Bernstein also found that there's a critical period during which regrowth must be completed or it will fail. After cutting the cords of a goldfish, he inserted Teflon spacers to block regeneration. The normal processes took place, but those, of course, the cells couldn't penetrate the divider. After the cellular activity had died down, Bernstein removed the barriers, but there was no further change. However, when he then cut off each damaged end, producing an even larger gap and re-injuring the cord, the cells started from scratch and healed the defects completely. Thus, there's good reason to believe that even long-standing spinal injuries can potentially be regenerated if we can extend the basic capabilities of human cells. One would expect to see some healing response in mammals, even if it fell short. After all, we only need the elongation and retachment of fibers, which does take place in peripheral nerves. Instead, the opposite happens. The cord cells die in a short distance above and below the injury. Cyst forms near ends, and instead of epidema, scar tissue fills the gaps. Only after this destruction is there an abortive attempt at regrowth. In humans, this amounts to only a few millimeters of elongated many months after the injury. By then, it's too late. The epidermal cells and nerve fibers can't penetrate the scar. Cysts and scars prevent cord regrowth in mammals. Why the difference between salamanders and mammals? The reason may lie in the cord's immediate response. In all animals, the injury instantly results in spinal shock, during which all neuronal activity is profoundly depressed. especially in the part of the cord still connected to the brain. Even the simplest reflexes disappear. As the shocks wear off, the cord below the injury becomes hyperactive. Its reflexes become different input of current. Now, its reflexes become tremendously exaggerated and lead to spastical paralysis of the muscles. An interesting difference in the duration of the shock in young salamanders and goldfish. It lasts only a few minutes, but it may endure for over a week in old ones. In mammals, it takes even longer to wear off, as 6 months in humans. Cow's experiment. It should be possible to cancel that polarity and replace it with growth stimulating -1 using a proper shaped electrode. Older injuries in which spinal shock has subsided might require a different input of current, as well as surgical removal of scar and cysts. The electrode material would have to be chosen carefully, for some metals are toxic to certain nerve cells. Also, humans have a low ratio of epiderma compared to spinal neurons, so we might have to add more. However, it should be relatively easy to culture more epidermal cells from a sample of patient's own and then inject them with when we put in the electrode. Not too many years ago, spinal accident victims usually died of infection or other complications quite soon. Now we can prolong their lives, but only at enormous social, financial, and psychological cost. Looking ahead, as in the case of heart damage, we now have hope for releasing regeneration in humans. Actually, the outlook for spinal regrowth is more promising. The cellular processes are more familiar, and there are a few groups like the American Paralysis Association that sponsor research for more imaginatively than government agencies. Thus, the electrical problems in spinal healing may be tackled sooner than in other fields. The public imagination has been captured by the computerized muscle stimulation techniques being developed by Gerald Petrovsky, an engineer at Wright State University in Dayton. The nationally televised sight of his patient, Nan Davis, and other paraplegics taking tentative steps and pedaling tricycles with their own muscle power was tremendously exciting. But if we can get the body to do the same things by itself, that will be even better. Any amount of regeneration would only make other techniques more effective. Even restoring 10% of lost function would be unimaginable blessings to those who are now helpless. I feel the electrical manipulation of spinal shock must be tested vigorously now, for this is perhaps the one area where the barriers of tragedy are closest to being broken. The brain. It might seem foolish to expect any regeneration in most complex of all biological structures, the brain. In salamanders, some fish, and most frogs in the tadpole stage can replace large parts of it, including optic lobes and the olfactory lobes, or forebrain, part from which our prized cerebral hemispheres developed in the course of evolution. Replacement depends on regrowth of remaining sensory nerves, the olfactory nerves, in the case of the forebrain, and the optic nerves for the optic lobe. When these nerves grow back into the area where brain has been destroyed, they stimulate the epidermal cells in the brain ventricles, which proliferates outward into the damaged part and then differentiates into new neurons and glial cells. If the animal's nose or eyes are removed so far that the injury zone receives no nerve input, no regeneration occurs. This brain regrowth begins much like that of limbs, with the connection of nerve fibers to an epithelial tissue. The epiderma, remember, is embryologically a close relative of the epidermis and, in fact, can be considered the central nervous system's inner skin. Since the electrical environment produced by a neuroepidermal junction is what stimulates cells into dedifferentiation and divide in the salamander limb stump. And since we started limb regeneration in the rat by crudely mimicking the signal, it seems likely that a similar stratagem could induce brain regeneration in animals normally lacking this ability. A form of shock called the spreading depression of Liao after its discovered neurologist A.A.P. Liao occurs after brain injuries. Starting at the site of damage, it extends in all directions until the entire cortex becomes electrically positive and all its neurons shut down. Liao studied it in response to small injuries when it persists for a few hours. We don't know whether it occurs in the salamander or how long it lasts after major damage to the mammalian brain. Concerted study of Liao's depression combined with the experiments in electrically stimulating the epidermal cells could open a way to self-repair of human brain. Amphibians can regenerate large parts of the brain. Recovery from stroke and head wounds taught us long ago that the brain has a great deal of plasticity. That is, it can reorganize so that undamaged regions take over tasks formerly done by the lost cells. Supplementation of this ability with even a small amount of regeneration might make recovery nearly complete for many brain damaged people. For the first time in history, neurologists can hope to progress from describing the brain and cord to mending them. As Jeffrey Reisman of London's Laboratory of Neurobiology recently reminded his colleagues, no immutable natural laws have been discovered that forever rule out repair of the nervous system.